good. It's good to see some familiar faces here. Hope everyone's doing well during the pandemic. And yes, Abhishek noticed some differences. So we'll wait another few seconds. Can everyone see my slides? Okay. Okay, should we get started? Okay, great. All right, so today I'm going to talk about two different projects that I've worked on with Elucidate over the years. We've been working together for quite a while now through different transitions. So I'll talk about some work that we did with poly and more pharmacokinetic-based measurements, and then some of the more recent work that we've done together as we move into spatial metabolomics using MALDI imaging. So I've spent a lot of time over the years developing different methods to try to measure metabolism in different ways. And so just as a brief highlight of what we've done in the past, and kind of we use a lot of the same methods now. We use stabilized token fusions into different mouse models of disease or cancer and rare disease. And we can follow nutrients based on using LC-MS measurements. And more recently, we moved into MALDI. So we showed that there were critical differences in lung tumors that were in the mouse model of cancer compared to when you took those same tumors out and grew them in culture. We showed that protein could be a nutrient supply for pancreatic cancer cells by making labeled yeast extracts or labeled yeast protein and feeding that to cancer cells. And then we went on and did that in mouse models where we made a labeled mouse albumin, and then did plasma exchange and plasmapheresis in mice to show that this also happens in vivo in pancreatic tumors. And this was all happening at MIT, and towards the end of my PhD, we started using a couple of different techniques involving this micro device, some toroidal microscopy, and finally MALDI imaging. And so I transitioned from MIT, and now I'm at Princeton. And when I came to Princeton, we decided to do, really pursue MALDI imaging much further and really try to do spatial metabolomics with this. So we have a Bruker Solarics XR with a 9.4 Tesla magnet, very cool FTI-CR mass spectrometer. And using this, we can make metabolite measurements with spatial resolution. So we can get to, near single cell, almost 10 to 15 micron. And as you can see, these are just brain sections. And so each one of these would be, basically an intensity plot of metabolites across the brain section, different metabolites. And in addition, we use conventional LC-MS and GC-MS. I'm going to talk about two ways that I've worked with Elucidata to answer questions using these methods. So the first is this project's exploring the pharmacokinetics of a novel compound for the treatment of cystinosis. So a brief background. So cystinosis is a rare inborn error of metabolism. It's a condition that's characterized by the loss of the lysosomal cysteine transporter. And when you lose this, cysteine accumulates in the lysosome and causes cell death. And some of the common organ sites are kidneys, eyes, and it's something that it's an inborn error, so it has to be treated. And one of the ways to treat it is using this drug called cysteamine. So cysteamine works. We know the mechanism of action in cystinosis, and so cysteamine enters a cell and enters a lysosome and reacts with cysteine to form a free cysteine here, which can be then exported by the cysteine transporter, and cysteamine disulfide, which can then be transported by the arginine transporter. But this is a story where, there's two things. So concentration really is everything. So you'll notice that the problem with cysteamine is the relatively short half-life. So, about you reach Cmax very early, and then it kind of comes down. And so if you can imagine being a patient with this disease, you would realize that you have to take, this drug, this non-enteric coated form, 6 times a day or so. And it's not a great process. It's a sulfur-containing compound. So I put the side effects here as vomiting, but there's a lot of other side effects. You can actually smell kind of the sulfur compound coming off of the patients. And so it's a story where, the idea would be if you could find a drug that would, both last longer and you could blunt the Cmax here to prevent these side effects from happening, that would be beneficial. And so when we were doing this, we had some novel compounds that we were testing, and we wound up doing this in rats and eventually in pigs. And we had several prodrug candidates, and we wound up amassing something like 1,200 samples, which required, absolute quantification, normalization through all of that. And so it was a lot of samples. And, for the first time, we also had a good look into understanding, what the possible consequences of cysteamine administration might be. So previously, a lot of efforts had focused on, delivering cysteamine and measuring the concentration of that, measuring the concentration of cysteine. But we're also interested in other applications where cysteamine has been proven to be effective, but had lesser known mechanisms of action. So some clinical trials using cysteamine in Huntington's disease, and it's not so clear that, cysteine is really the target there. So we had this major problem where we had all these samples, and we're doing absolute quantification of something like 50 metabolites where we make standard extracts of these, and we needed a solution. So with that, we turned to Lucidata, and we used and helped kind of inform QuantFit, and a lot of feedback and kind of a lot of going back and forth on different features. And so this was a really good application. And so what this dashboard allows you to do is you can normalize to internal standards. You can auto quantify all of these metabolites using these curve fits. So in cases where things aren't linear, and then you can manage all of this stuff very easily in a way that would be very difficult and without any bioinformatics. So this was extremely helpful and a good use case in terms of throughput and in terms of just really working together on a really interesting biological problem. And so we're actually moving that drug ahead. As I mentioned, it's in pigs now, and so we have that data, and we hope to be in humans within a year. So the problem, though, with some of these LC-MS measurements is that you grind up the tissue, and so you're left with just these bulk measurements. And so we wanted to develop MALDI imaging mass spectrometry to get these spatially resolved measurements. And I'll focus on some data from the kidney. But we have all these emerging techniques here that can measure kind of single cell omics, genomics, transcriptomics. But if you notice our traditional methods in metabolomics, you need many cells. And if you want to study a specific cell type, there's some clever work from Matt Vanderheiden's group using FACS and kind of these long-term labeling studies. But in general, FACS over the period of sorting, a lot of these metabolites are changing, as we know. So if you want to study cell type specific metabolism, it's relatively difficult. And so we kind of needed a better way to do this. So, and as we know, the challenges for the metabolic assays, in contrast to some of the sequencing-based methods, is that you can't really amplify metabolites. The chemical complexity is quite high, and there's a wide concentration range. So yeah, the question really is, how can we make these meaningful metabolic measurements in heterogeneous tissues? And so we've been using MALDI imaging mass spectrometry. And so briefly, it looks like this. So you have your tissue coded on a slide, which is represented by analytes, and then you have a matrix that goes along with this, and then you have a radiation by this UV laser, and that causes desorption, and you get desalvation, and you send that to the mass spectrometer, the metabolite ions to the mass spectrometer. And so The biggest problem we encountered initially, and again, this is the point where we reached out to Elucidata, was that we're generating data sets now with imaging mass spectrometry that are 100 to 200 to 300 gigabytes large. So every single pixel is its own mass spectrum. So it's almost like an individual sample on LCMS. And there's all these steps that Elucidata has worked on for LMaven. So it's kind of the same problem through pre-processing and peak picking. But what I was really impressed with is that there wasn't much experience in working with a lot of these different image overlay pipelines and kind of integrating these other data sets, and they were willing to try and put many people on the team. So Kailash, Shafali. I think Shafali must have a clone because I've heard her mentioned in pretty much every talk today. So Shafali, if you know how to clone yourself, let me know. And so, yeah, I was really impressed with kind of the versatility and adaptability, basically, of the Elucidata team to try to tackle this problem, along with these multivariate analyses that include clustering and segmentation. So I'll show you some examples of that as well. And so this is another slide. This is an example of some of the imaging mass spectrometry data that we're getting, and also an example of how, as you've heard today in many talks probably, looking at metabolomics versus kind of relative flux or relative contribution. So this is one of my favorite examples from imaging mass spec so far. So if you take a section of a kidney, you can look at the cortex and the medulla. If you just look at labeled malate, or if you look at unlabeled malate, you can see the concentration in the cortex and the medulla is relatively consistent. If you quantify it, there's no statistical difference. But if you look at the relative contribution, malate becomes much more interesting. So, if you just did metabolomics, you wouldn't necessarily get some of the interesting information. So we're looking at metabolomics and kind of flux. And malate becomes much more interesting when you start to ask, where does malate come from? So here we have the unlabeled malate, which is relatively consistent in the cortex and the medulla. But if you ask where malate comes from, so we're asking how much glucose labels malate when you infuse glucose into the mouse kidney, how much acetate contributes to malate, how much 3-HB contributes to malate. And so you'll see that for these three tracers that we infuse into the mice, there's not much difference in the cortex and the medulla in terms of relative contribution of these nutrients to malate. But then we start to see some really interesting differences. So, for example, we see cortex-specific contribution. If you take the most extreme example, you can look at something like citrate, and you can see that citrate is creating labeled malate in just the cortex. And so, and that's for the reason that the cortex actually expresses a citrate transporter. So we're interested in also going forward and using kind of an integrated omics type platform on MALDI imaging mass spec data to understand when enzyme expression correlates with metabolic activity. And then we can see another example where we have kind of fatty acids contributing specifically in the medulla. And then we can use glucose and glycerol and show contribution and relative fluxes through glycolysis and gluconeogenesis, which we've done. And so we have this for most mouse tissues now in kind of the normal state, and then we're working through different disease contexts, different cancer contexts, and Huntington's. So what I really enjoyed about working with the Poly team on this project, especially and going forward, is these custom solutions. They really have enabled us to manage these large data sets and come up with a good way to look at it and visualize it. It's hard to find kind of software engineers with diverse scientific backgrounds and expertise in biology, and this team has been really great, and I've worked with a number of people over the years at Elucidata. It would be too difficult to name everyone, but it's been a really great experience. And I'll end on another slide that we generated together that's kind of thought-provoking and one of my favorite examples. So when we took this and we did dimensionality reduction, we took these imaging mass spec data sets and we did dimensionality reduction. If you look on the left, we have this metabolic classifier that we used. And on the right, you have the Allen Brain Atlas. And when I went into this experiment, I thought we might get clustering of metabolite signatures based on cell type. Maybe all neurons and would do one thing, and all astrocytes would do another thing. It was really fascinating that you get these clusters that are representative of anatomical regions, and so this was pretty striking. And we're following up on this. This has been shown for other things like proteins and lipids that might work, but based on that the brain is usually just burning glucose, I wouldn't have expected this signature to come from small molecules necessarily. So we're trying to figure that out. But this gives you a good sense, and we can do this in the normal and disease context. So you can imagine if you have a mouse model of disease or other that you could compare, and you can kind of do the clustering. And so we've done that with Elucidata with some Huntington's disease mice, and continuing to work on these things. So with that, I'd just like to thank the Elucidata team for being available. The other great thing I can mention is they've really interacted with the people in my lab quite well, and my graduate student has benefited a lot from working with them, and the availability has been awesome. So thanks again, and thanks to Abhishek for inviting me to this talk, and I'd like to thank my funding sources as well. And I'd be happy to take any questions about imaging mass spec or anything else.